Hi guys, welcome back to the channel. Today we will be reviewing some IB style test topic 7 questions for the paper 1. The official IB question bank cannot be shared, that's kind of illegal, so we're doing the IB style test questions from InThinking. This is a PDF you can find on their webpage, and today we're going to solve the questions together. So I will read the question, I will give you some seconds to pause and try to solve it by yourselves, and then I'll solve it out loud and explain my reasoning. Let's get started. Alright, let's read the first question. The X-ray diffraction pattern obtained by Rosalind Franklin indicated that the structure of DNA had A. Complementary base pairs, B. A helical structure, C. A backbone, or D. Nucleosomes. Okay, I'm going to give you a second to pause and practice the question, and we'll be back. Okay, let's see. Based on the theory, I can deduce that the correct answer is B, a helical structure, and I am also able to deduce this, so if you don't know the correct answer, let's use a little bit of logic. So, nucleosomes is the first one to be discarded because Rosalind Franklin used a DNA molecule, and nucleosomes are used to pack DNA, so the X-ray crystallography would not have had the same results. So nucleosomes, it's not D. Then a backbone. The backbone of the DNA are the two strands. So it can show that it has a backbone, but the models for the DNA molecule were invented later by Watson and Crick. So backbone, it's not it. And the complementary base pairs is not able to be seen in this picture, so it's also not A. So I believe the answer would be B. Let's move on to the second question. So 2. The structure of a ribosome includes 1. Ribosome RNA, 2. Protein, and 3. Enzymes. So we have the options: only ribosomal RNA, ribosomal RNA and proteins, RNA and enzymes, and all of them. Okay, now you have time to pause. I believe that the correct answer is B, only ribosomal RNA and proteins. Let's review our ribosome structure. So first of all, we have our mRNA, right? This sequence of codons. The sequence of codons enters the ribosome, and under it we have the small subunit, and above we have the large subunit. Within the large subunit, we have the E, P, and A sites, which are tRNA. We have this tRNA here, and from the tRNA, there is a protein yielded thanks to the translation process. So this is our ribosome. We can identify here that we have a large subunit and a small subunit made of proteins, so that was option 2 from the question. And then we also have, we need to notice it is theory, that these two subunits are held together by ribosomal RNA. So we have option 1. Enzymes, however, are not an intrinsic part of the ribosome. They can be associated to this RNA and to the ribosome in order to be able to produce other compounds, but it's not part of the ribosome. So we have ribosomal RNA, and we have proteins, but we do not have enzymes. So the correct answer is B. 3. The role of DNA gyrase is to A. Rewind DNA after transcription, B. Keep the DNA strands separate to allow base pairing, C. Alter DNA bonding to allow helicase to unwind the DNA, or D. Form covalent bonds in the DNA backbone. It's time for you to pause and try to guess for yourself. Let's try using logic again. We know that DNA gyrase is used in replication, so rewinding DNA after transcription would definitely not be the correct answer. Option B says keep the DNA strands separate to allow base pairing. That is what the single-stranded binding proteins do. So that is not DNA gyrase's job. And lastly, D that says form covalent bonds in the DNA backbone. This is incorrect because the covalent bonds are formed due to the physical and chemical properties of the compounds that form the DNA backbone, so DNA gyrase is not needed for that. So the correct answer would be C. Alter DNA bonding to allow helicase to unwind the DNA. So helicase unwinds the DNA and it forms this replication fork, and there is a kind of stress formed behind helicase. So the DNA gyrase is placed next to the helicase in order to modify chemically the DNA so it's easier for it to be unwinded. We're in question 4 now. Which of the following do not occur after transcription in prokaryotes? 1. Attachment of mRNA to a ribosome. 2. Removal of introns. 3. Movement of mRNA through the nuclear membrane. And our options are only the third, the first and the second only, the second and the third only, and all of them. So I'll give you a second to think about this question. Okay, this one is an easy peasy one. Let's begin with the first annotation. It is that the movement of mRNA through the nuclear membrane. In prokaryotes, we do not have a nuclear membrane because we do not really have a nucleus. That's the meaning of prokaryotes in contrast to eukaryotes. So we can say that 3 is going to be in the answers. So if 3 is going to be the answers, B is automatically ruled out. Then we know that prokaryotes do have ribosomes. So this one should not be in the answer. So again, D can be ruled out. So now we have to decide if it's either A or C. So we have to remember what introns are and what is their purpose. Introns are non-coding regions of the DNA sequence, and these are only found in eukaryotes, so prokaryotes do not have them, so 2 is correct. And basically the correct answer then would be C. Let's move on to question 5. A quaternary structure is found in some proteins because they have A. Many amino acids, B. One polypeptide chain, C. More than one polypeptide chain, or D. Enzymatic properties. So as we can see here, we are talking about structure. So we will review the 4 levels of structure of proteins. The first level, level 1, is the primary structure. It is the sequence of amino acids. So if they put you valine and then another amino acid, and so you just have a chain of amino acids, that will be the primary structure. Then the secondary structure is how the hydrogen bonds between these amino acids interact with each other without taking into account the carbon groups. So you don't worry about that. You have to know that it's the interaction between the hydrogen bonds. So we can either have an alpha helix, which looks something like a coil of this type, and the beta plates, which are represented with arrows. So the interaction of these ones, that is, for example, the secondary structure of a protein. Then the tertiary structure is a three-dimensional model of this protein, and however it shows hydrophobic interactions. For example, let's say like this is a protein, and in here we have a hydrophobic bond. We have compounds which are hydrophobic, which are inside the protein, so it shows the three-dimensional structure. And the quaternary structure is when different polypeptides interact within each other. So primary structure, amino acids. Secondary structure, the beta plates, which are like this, and the alpha helix. So yes, the beta helix is the beta plate, and that shows the hydrogen bonds within a polypeptide. The tertiary structure, which is a tridimensional structure which shows hydrophobic interactions and, for example, disulfide compounds that are within the protein. and, for example, other intermolecular forces, the van der Waals forces, and a quaternary structure is the interaction between polypeptides. So let's solve this question now that we know that. Quaternary structure is found in some proteins because they have many amino acids. That is valid. One polypeptide chain. That is incorrect because we know that quaternary structure is the interaction between polypeptides. More than one polypeptide chain. That is correct. And lastly, enzymic properties. That would be incorrect because the quaternary structure, the interaction of the polypeptides can be for any protein. It is not necessarily an enzyme. And some enzymes have quaternary structure, but some don't. Some just have a tertiary structure. And so it is not like the reason for a quaternary structure existing in the proteins. It's the cause of why a quaternary structure exists. So there being many amino acids is not the reason why they have a quaternary structure, because the reason for having a primary structure is also having many amino acids. So the main reason is having more than one polypeptide chain. Question 6 is very long, so I'm going to leave you to read it. Just pause if you want to, and now I will analyze it. Okay, so what this question is asking is which of the following sequences of events correctly describes tRNA activation. First of all, what we need to know is what is tRNA activation. Okay, so first of all, to solve this question, we need to know that tRNA needs to be activated because tRNA is in charge of the formation of new peptide bonds, and this requires energy. So tRNA needs to have enough energy to produce this polypeptide. We're not going to lose time reading everything because this is a long question. The correct answer is D. We will focus on that explanation. So the first step is that ATP and a specific amino acid bind to tRNA synthase. So we know that an enzyme is involved, and ATP, which will give energy to the tRNA molecule, binds to this enzyme. This amino acid is activated using the ATP. So we have our enzyme, ATP enters an amino acid, and ATP gives energy to the amino acid, and then the tRNA with the correct anticodon binds to the synthase. So firstly, we only have the ATP and the amino acid, and then the ATP gives energy to the amino acid, and the tRNA, a specific tRNA, binds to this tRNA synthase, and the amino acid is attached to this tRNA. It gives this energy to the tRNA and then leaves the enzyme. So the tRNA can now be activated. Question 7 states the following. The formation of major mRNA after transcription involves A. RNA polymerase, B. Removal of exons, C. Splicing, and D. Ribosomes. Okay, I'll give you a moment to pause this question. Okay, let's start with the process of deduction. The first option we have is RNA polymerase. This is involved in the process of transcription, so it could be the correct answer. However, RNA polymerase is used to bind nucleotides into the replication fork, so this is not used in maturing messenger RNA. Let's move on to B, the removal of exons. Exons are the opposite of introns. If introns do not code for any information, then exons are the part that codes for information. They should not be removed, so the removal of exons is not the correct answer. Splicing sounds like the correct answer because splicing is the name of the removal of introns. It is a process called splicing, so this could be the correct answer. And then the ribosomes. We know that ribosomes are used in translation, not in transcription, therefore this is not the correct answer, and the answer is C. Question 8. The role of the A site on the ribosome is for A. Forming a peptide bond, B. Removal of the amino acid from the tRNA, C. Causing the assembly of the large and small subunits of the ribosome, or D. Binding the mRNA codon to the tRNA anticodon. I will leave you to think about this question. There are three sites on a ribosome: the A site, which is the amino acid site, the P site, which is the peptidyl site, and the E site, which is the exit site. The A site is the one we're assessing right now, and as it is called the amino acid site, we know that it has something to do with amino acids. So we can discard C, because causing the assembly of the large and small subunits of the ribosome must not be a function of the A site. In fact, we talked before about it, and it is the function of the ribosomal RNA. So we have forming a peptide bond, and this is incorrect because the peptide bond is the function of the P site, because it is the peptidyl bond. So A is not the correct answer. So now we have the removal of the amino acid from the tRNA and binding the mRNA codon to the tRNA anticodon. B does not really make sense at all, because what we are doing is creating amino acids and creating peptide bonds. And so removing something that the tRNA has created is not a function that the A site can perform. So the correct answer is D. In fact, the mRNA arrives to the A site, and when the A site detects this starting codon, it moves on into the P site. So what it does is basically uniting the mRNA codon to the tRNA anticodon. And the correct answer is D. Question 9 is, non-coding regions of human DNA can be 1. Repetitive sequences, 2. Regulations of gene expression, 3. Transcribed into mRNA, and 4. Telomeres. And here we have all of our options. I'm going to let you think about this question for a second. Okay, let's analyze this question together. First of all, we have repetitive sequences. They can perfectly be repetitive sequences. So 1 would be okay. 2. Regulators of gene expression. It can be a regulator of gene expression, because imagine you have a non-coding region, then that which you wanted to code is silenced. So now you are regulating gene expression. So we can discard A, because we know that 2 is correct. Then 3. Transcribed into mRNA. We explained before that this does not happen, because mRNA in eukaryotes, because we are talking about human DNA, is matured, and the introns, the non-coding regions of the human DNA, are taken out, are spliced, as one says. So 3 is not correct. So D is also discarded, and now we have to see if number 4 is true, so we can choose between B and C. Okay, telomeres. This is correct, because telomeres, if you don't remember, are this part you have a chromosome, well, 2 sister chromatids, and in these parts we have our telomeres. It's at the edge, and this is very useful for organisms, because during the process of replication, transcription, and everything, some part can be damaged or lost, or not all of it is transcribed because of the amount of energy it needs to be used. So these telomeres are non-coding regions, because if they are lost, they were going to be lost anyways, probably due to splicing, so it doesn't harm the actually coding region of DNA. So 1, 2, and 4 are the correct statements, because this is what non-coding regions are. So the correct answer is B. Question 10 says, bioinformatics applications in databases containing information on DNA-based sequences can be used to A. Identify homologous sequences in different species. B. Identify infectious diseases. C. Derived 3D protein structures. Or D. Sequence of a genome. Okay, this question is not only very easy to answer if you know the theory, but it is also kind of common sense, so we can just check out some things and discard some answers. For example, C, derived 3D protein structures, is incorrect. With only the base sequences, we cannot derive the 3D protein structures, because we don't know the interaction between the particles. Then the sequence of a genome, also, it doesn't really make any sense. Identify infectious diseases. Let's revise the difference between contagious and infectious. Contagious is when you have a pathogen, an antigen that can be transmitted from one organism to another organism, it can be contagious. And infectious is when you have this pathogen attack an organism. But the key is that an infectious disease is not necessarily contagious. For example, I can get food poisoning, but then that food poisoning can be not contagious. So that's the difference. And how do you identify this? Well, I don't see how you would do it with DNA-based sequences, because how you determine a disease is infectious is through experimental testing, I guess. So this would also be a no, and mostly because the one that makes the most sense that you would use bioinformatics for is to identify homologous sequences in different species. This is to see, for example, if a cow and a monkey have any homologous sequences in the DNA-based sequences, to determine, for example, similarity, or for the study of cladistics. Cladistics, to do like a clade diagram, which is how the species have been separated through time. This is time, and how they diverge. So that's what bioinformatics and DNA-based sequences in bioinformatics can be used for. So I would say A is the correct answer. Okay, so this was all for today's video. Thank you so much for coming, and if you like it, just leave a like, subscribe, and leave a comment if you need any other videos. This is for the paper 1 topic 7 questions. For the paper 2, you can go to the reproduction list in my channel and you will find it there. These are short paper 2 questions. Also IB style, but not official IB. Thank you so much. Bye.